The goal of a clonogenic assay is to see how many cells continue growing and form colonies after treatment. It is useful for testing cancer therapies where it complements immediate cell toxicity with later effects of your treatment. In other words, do cancer cells survive and continue to divide, or was it a total annihilation? A listing of all the equipment needed may be seen in the description below. Key to success is careful preparation, so be sure that all the tubes, wells, and vials are marked with the treatments and controls that you will be using during this treatment. Before performing the assay itself, you'll need to treat the cells. In this example, we are treating our cells with a scorbate. Following the treatment, remove the media and rinse. Add 500 microliters of 0.25% trypsin EDTA. Your volumes may differ if you are not using a 60 mm dish, as used in this example. Once treated, rinsed, and washed, return the cells to the incubator to detach the cells. While the cells are detaching, add 1.14 milliliters of full media to the Epub tubes you will be using, 4 in our case. Give the cells approximately 5 to 10 minutes to detach from the treatment plates. Remove the 60 mm dishes from the incubator and add 500 microliters of full media to each dish. Give them a good rinse to spread the cells evenly throughout the media. With your 1 mm pipette, add approximately 50 microliters of cells to the Epub tubes. Mix the solution in the Epub tubes thoroughly and add 1 ml of this solution to the 9 ml of isotone in a coulter counter vial. To avoid confusion, each Epub, counter vial, plate, and well that you are using should be clearly marked. There is now a 10:1 ratio of solution in the coulter counter vial. Place the counter vials in the cell counter, record the values, and return to the hood. Now that you know the cell concentration, you can proceed with the plating step. Back under the hood and using a clean pipette, draw out 900 cells and add to the 12 milliliters of media that you had earlier placed in the conical tube. Mix the solution with your pipette and transfer to 6 well culture plates. In this example, we plated 3 wells of 4 milliliters each. Now that your plating is complete, return the cells to the incubator and wait 10 to 14 days for the colonies to grow large enough to easily count. Once the colonies have grown large enough to count, it is time to stain them. We're using Comasi blue. Begin by removing the cells from the incubator and suction off the media from all the plates. Rinse with ethanol, spraying on the side of the wells to avoid potentially disrupting the colonies with the force of the spray. Place the trays flat and add a little ethanol and allow it to sit for 5 minutes in order to fix the cells. Suction off the ethanol and place the trays on a flat surface. Add the Comasi blue to each 60 mm well and cover to prevent the evaporation of methanol in the solution. Allow to stain for 15 minutes. Notice that once again, each of the trays is carefully labeled. After the elapsed time, siphon off the excess Comasi blue, then rinse each tray under running water until all the excess dye is washed off. We rinsed 3 times. Allow to air dry before counting. Thanks for watching.